All right, let's see how quickly we can get through mutations and repair. So initially, there's going to be some wild-type DNA sequence, the most common sequence within a population. And then at some rate, there are going to be changes, whether they were errors during replication or spontaneous chemical reactions or mutagens. Now, here's where the arms race comes in. So there are lots of mechanisms or machinery that are going to try to repair that change. Now, this machinery doesn't care if that change is neutral or could potentially be a good or bad change for the cell. It's going to detect an error and try to fix it. Now, if the DNA replication machinery moves on before that DNA and the sort of cell division machinery moves on before that change can be repaired, then it's going to be incorporated into the cell as a mutation. And so now that change in DNA is going to be passed on either to daughter somatic cells or potentially to progeny. Okay, so now we want to talk about how we describe these changes based on different perspectives. So from the DNA, protein, functional level. So we talked about really three different DNA mutations that can occur. We can have point mutations, which is also called a base substitution, insertions, and deletions. So if we look at this original sequence, see if you can write out an example of each of these types of DNA mutations based on the original sequence. So these are the examples from the figure in the textbook where we have a point mutation, a base substitution of T to C, which is a type of transition mutation. Here this T has been inserted, and here the original T was deleted. Depending on the nature of insertions and deletions, there can be sort of different effects. So there can be expanded or contracted nucleotide repeats, and I showed you some examples of those in human disease. The location of those repeats matters. So sometimes they'd have no effect. Sometimes they are in a regulatory sequence or intronic sequence or exonic sequence. It could be in the UTR or an actual coding sequence. So related to that, if the insertions and deletions are in protein coding sequence, and not in factors of 3 base pairs, which essentially would insert or delete specific codons, this could lead to a frame shift mutation in the protein coding sequence. Frame shift mutations just change the reading frame or the codons downstream of the insertion or deletion. So again, if you look at the wild type sequence, we have these codons, and downstream of the insertion, we've now changed that codon, and we've changed the codon at the deletion. And again, it's going to be downstream. So from the protein perspective, these DNA point mutations and insertion and deletions can lead to changes in the protein coding sequence. So the first change can be a nonsense mutation, where a codon for an amino acid is changed into a stop codon. So again, this could arise from a point mutation or an insertion or deletion. A missense mutation is where the amino acid changes. So the codon for one amino acid is changed into a different amino acid. Again, this can change into an amino acid of the same chemical properties, which might not have that big of an effect, or it could be a totally different amino acid that causes some serious problems with the protein function. Then finally, because of redundancy in the genetic code, there can be a point mutation that leads to a silent mutation, where an amino acid encodes for the same amino acid, or a codon encodes for the same amino acid. Even though it's been changed, multiple codons can encode for the same amino acid. Now, usually silent mutations are the least harmful. Missense are something in between, depending on the nature. And the nonsense mutations, again, depending on where it is in the protein sequence, is going to be the most disruptive, right? If it's an early stop codon, then you're pretty much not making the protein at all. Then describing the phenotype might be more challenging because it's a little more, there's more ways that we can talk about the effect of a mutation. So typically, forward mutations are actually creating the mutation itself, and reverse mutations are going back to wild type. LOF is referring to loss of function, and this is where the wild type function of the protein is now no to little function. Gain of function, GOF, is a little different. This is where there's been a mutation and now it has a new function, or it's functioning when it's not supposed to, which is essentially giving it a new function. So for instance, there could be a change in a sequence that gives an enzyme maybe a new substrate that it binds to, or it could be that an enzyme that's supposed to be working in the eye now works in gut cells. That's a new function for that protein. Conditional mutations are, or phenotypes are only mutant at specific conditions. So maybe temperature, pH, nutrition needs, stuff like that. Lethal mutations obviously go from lethality, right? Like killing the cell or the organism when that mutation is present. Again, silent mutations at the phenotype level just mean that it has a wild type function, even though it's been, there's been a change. And then finally, a suppressor mutation is where there has been a mutation that leads to some effect, whether it's loss of function, gain of function, conditional, lethal, whatever it is. That mutation exists and is affecting the phenotype. There is a mutation in an additional gene that leads it back to being wild type. So it's now a different kind of wild type. So again, the key here is that the mutation happens in a different place. It doesn't have to be a different gene, right? Because we saw intragenic suppressors. Regardless, it needs to be a different mutation. Oops, that didn't work. It needs to be a mutation somewhere else, whether it's in the same gene, which is an intragenic suppressor, or a different gene, intergenic suppressor, that's taking that phenotype back to wild type. And again, these are important because they reveal genes that are working in the same pathway, but they can also reveal sort of the nature of a mutation within a specific gene. Okay, so that's kind of a lot. I'm going to stop there and I'll make a different video for repair.